Good afternoon, everybody. I am Francesco Zangaro, and here I present you our work about the gap analysis of non-indigenous species occurring in the Mediterranean Sea DNA barcodes in the reference libraries. The key points of our presentation are, of course, the limitations of the conventional taxonomic-based tools, the molecular tools viewed as a solution to these limitations, and an analysis of the advantages and limitations of the molecular tools as well, the true objectives of our work, material and methods that we used, our results, and some final considerations. Until the last years, the identification of species has been carried out only through visual methods, mainly based on dichotomous keys that are generally based on recognizable adult features, as well as on particular body parts like the gonads. Therefore, the investigations based on traditional sampling and taxonomic-based tools show nowadays many limitations. In fact, the visual methods can easily fail the identification of pre-larval stages, larval stages, and juveniles, and of these individuals whose morphology was altered due to stressful environmental conditions, specimens that were damaged when they were sampled, and specimens that were not preserved in the proper way, resulting again in damaged specimens. When looking for rare or cryptic species, like for instance the non-indigenous species that are recently entering the Mediterranean Sea, these methods show even more limitations. Nevertheless, the traditional taxonomic-based tools involve the sampling of specific individuals and the expertise of taxonomists for different taxonomic groups that could fail in the identification of rare and cryptic species. Also, to be able to identify recently introduced species, taxonomy experts from the original area of these species are required, and it can be something hard to access when the original area of an unknown sample is incognita. Indeed, to have a consistency of biological records, mainly when talking about non-indigenous species, it is fundamental to assess the distribution of these species and their range expansion. Nevertheless, the urgency of monitoring the occurrence and the distribution of non-indigenous species is unanimously requested by the Mediterranean scientific community. A promising and innovative approach for the species identification in the biomonitoring of the ecosystem is the use of DNA barcoding and metabarcoding, a recent and revolutionary concept that refers to a single species identification with the use of a short fragment of DNA. Especially, its application on the identification of species at low densities can prove to be very profitable. The direct consequence of this concept is that another innovative method is the use of the environmental DNA, used to identify the source of the DNA extracted from environmental samples, such as water or sediment. Therefore, the use of environmental DNA comes out to be an efficient and innovative technique for identifying the presence, the distribution, and the spread of non-indigenous species throughout the Mediterranean Sea, avoiding all the difficulties, limitations, and costs that generally appear through the application of traditional sampling approaches. Nevertheless, this is a non-disruptive sampling technique. However, even the application of molecular tools faces right now some problems and some limitations. Currently, one of the main limitations in the use of molecular tools for species identification is the incompleteness of the DNA barcode reference libraries. The most commonly used barcode reference libraries are GenBank and BOLD systems. Both these databases provide a global list of species with several data elements. For having a perfect efficiency, Using the molecular tools, all the species on Earth should be identifiable through their DNA barcodes, but the current condition of the reference libraries is totally incomplete. This leads to an actual inefficiency in using the molecular tools, including the environmental DNA, for investigating the occurrence, the distribution, and the new records of non-indigenous species in the Mediterranean Sea. Nevertheless, the success of the molecular techniques and the applicability of the environmental DNA is based on the efficiency of the primer sets on the largest number of tags as well. For these reasons, the main aims of our work are to assess how many non-indigenous species occurring nowadays in the Mediterranean Sea can actually be identified through the environmental DNA, and to assess how many public primer pairs are actually available for the barcoded NIS. This leads us to promote the transnational, transcontinental, the global cooperation for reducing the current gaps. We started from the most updated checklist of confirmed alien species occurring in the Mediterranean Sea, assessed using the Inventory of Non-Indigenous Species at the start of 2020, published by Zenetos and Galanidi. Here, a total of 666 marine non-indigenous species established in the Mediterranean Sea are listed, resulting in the most complete checklist about the knowledge regarding the non-indigenous species presence in the Mediterranean Sea. The species were divided into 10 high-ranked taxonomic arrangements, themselves divided within 133 orders. Each of the 666 non-indigenous species retrieved has been searched on the two main DNA barcode reference libraries, both Systems and GenBank, checking for the presence of the barcode. Later on, for the species whose barcode was available, we examined if they publicly show the primer pairs that had been used for the gene amplification process. So, how many non-indigenous species occurring in the Mediterranean Sea can actually be identified through the environmental DNA? The answer is 449 species out of 666 non-indigenous species, corresponding to 67.42% of the non-indigenous species occurring in the Mediterranean Sea. This means that there is a gap in the DNA barcode reference libraries corresponding to 32.58%. Something that has to be highlighted for its importance is that each of the 449 barcoded non-indigenous species can be identified through the investigation of the mitochondrial cytochrome C oxidase subunit 1 gene. More in detail, here we show you the gap for each of the 10 groups retrieved. For fishes, 89 non-indigenous species and 15 orders were retrieved, with a gap of barcoded species of 3.37%. For bryozoa, 30 species and 2 orders were retrieved, with a gap of barcoded species of 76.67%. For parasites, 25 species and 10 orders were retrieved, with a gap of barcoded species of 56%. For phytobenthos, 112 non-indigenous species and 26 orders were retrieved, with a gap of barcoded species of 17.86%. For Ascidians, 26 non-indigenous species and 3 orders were retrieved, with a gap of barcoded species of 15.38%. For Crustacea, 83 species and 7 orders were retrieved, with a gap of barcoded species of 31.33%. For Miscellanea, 40 species and 21 orders were retrieved, with a gap of barcoded species of 20%. For Mollusca, 156 species and 28 orders were retrieved, with a gap of barcoded species of 61.28%. For Polychaeta, 65 non-indigenous species and 7 orders were retrieved, with a gap of barcoded species of 44.62%. For zooplankton, 38 non-indigenous species and 14 orders were retrieved, with a gap of barcoded species of 26.32%. Lastly, as we said, for each of the 449 barcoded species, the availability for public primary pairs has been investigated. Since all of the retrieved species can be identified through the COI gene, the investigation has been carried out on COI records. What comes out is that 249 out of 449 barcoded non-indigenous species do not show any publicly available primary pair set, corresponding to a gap in the availability of primary pairs of 55.46%. In particular, for animal non-indigenous species, no publicly available primary pairs has been retrieved for 398 species, corresponding to 70% of the retrieved animal species. However, the 64% of the species that showed primary pairs can be investigated using the highlighted 4 primary pairs. For plant and algae non-indigenous species, no publicly available primary pairs were available for 68 species, corresponding to 64.76% of the retrieved vegetable non-indigenous species. However, the 70% of the species whose showed primary pairs can be investigated using the 4 highlighted primary pairs. So, for resuming, the 32.58% of non-indigenous species occurring in the Mediterranean Sea cannot be identified through the use of environmental DNA, and 55.46% of the non-indigenous species that actually can be identified through the use of environmental DNA do not show any specific publicly available primary pair set for the amplification process when using COI as the target gene. These results highlight the importance of analyzing at a regional, national, and transnational level the barcode gaps in the reference libraries for the future application of DNA barcoding and metabarcoding in regional, national, and transnational biomonitoring assessment. This study reveals the current gaps existing in the DNA barcodes and primary pairs for the non-indigenous species already established in the Mediterranean Sea, but further studies need to be carried out all around the globe to ensure a higher completeness of the DNA barcode reference libraries to prevent new invasions by new non-indigenous species in the Mediterranean Sea, allowing the early assessment and identification of new non-indigenous species that can occur in the Mediterranean Sea. Thank you for your attention, and we are here to answer all your questions, and we wait you to the Research Center for Fisheries and Aquaculture of Acquatina di Frigole at the University of Salento in Lecce, in Italy. Thank you.